This is Dr. Sonia Shetty from Department of Microbiology, KG Somaiya College of Science and Commerce. Today, we are going to see the mode of action of antibacterial agents on bacteria. Antibacterial agents can be broadly divided into two types of agents, that is bacteriostatic agents. These are the agents who inhibit bacterial growth but do not actually kill the organism. So when the bacteriostatic agent is removed from the environment of the bacteria may again start growing. Under this you have many antibiotics. Some of the examples are chloramphenicol, erythromycin, trimethoprim, sulfonamides and tetracyclines. On the other hand, bactericidal agents are those who actively kill the organism. In these also there are many antibiotics under this group. Those are the aminoglycosides like streptomycin, the beta-lactams, that is the penicillins, vancomycin, ciprofloxacin, rifampicin and metronidazole. So coming to the mode of action of these antibacterial agents, now these agents kill the bacteria or prevent their growth by a variety of ways. They inhibit many important cellular processes. So we can broadly put them into these 6 different categories. The first are those antibacterial agents who inhibit cell wall synthesis, that is peptidoglycan synthesis. The second category are those antibiotics who will inhibit cell membrane synthesis. Third, antibiotics that prevent synthesis of proteins. Fourth, antibiotics preventing DNA synthesis. Fifth, agents that prevent RNA synthesis. And finally, there are few antibiotics which have other modes of action which may target some other important metabolic pathways. Now we all know how a bacterial cell looks like. Now in this topic today we are going to discuss only those agents which are active against bacteria. So we are not talking about agents that kill fungi or viruses or other life forms. So all these are only the ones which are affecting bacteria. So this is the structure of a common prokaryotic cell of a bacterium, and we can see in the center over here the DNA, the chromosome of the bacterium. We can see that the chromosome can undergo, or the DNA can undergo transcription to give you formation of mRNA. mRNA can undergo translation, and you can see here that is formation of protein that is happening with the help of ribosomes. Here you have another important biochemical pathway happening, that is folic acid synthesis. Also in this structure we can see two important parts of the bacterial cell, that is the cell membrane on the inner side and the cell wall, that is the outermost covering of the bacteria. So let us start with different categories of these antibacterial agents. So first are those agents which inhibit synthesis of the cell wall of the bacterium. Now we all know that bacterial cell wall consists of peptidoglycan. So these agents prevent the synthesis of peptidoglycan. Some of the examples which are put up over here are the beta-lactams, the glycopeptides, cycloserins, bacitracin and others. So peptidoglycan is an important part of the bacterial cell. We know that gram-positive bacteria contain a very high concentration or a thick layer of peptidoglycan compared to gram-negative, and because of this, the effect of these antibacterial agents on bacteria can vary depending on their gram nature. Also, an important part or important point of this antibacterial agents is that they are non-toxic to humans, because human cells do not contain peptidoglycan. So agents which affect peptidoglycan synthesis become very specific to only bacteria. So in a way that is an advantage of these antibacterial agents. So we are seeing the different classes of antibiotics under this. The first are the beta-lactams. Now the beta-lactams consist of few more subclasses within them. The first are the penicillins over here. We all know the story of how penicillin was discovered. It was the first antibiotic to be discovered, and it is made by penicillium fungi, and it is effective against majorly gram-positive organisms. So under penicillin there are many different types, that is penicillin and derivatives of penicillin, that is penicillin G, penicillin V, carbapenicillin, cloxacillin, methicillin and oxacillin. So these are all the beta-lactam antibiotics. The second subclass under this are the CFMs. Examples of CFMs are cephalothrin and cefotaxime. Another class of beta-lactams is the penins. Under penins, there are examples like carbapenem and sulfopenems. All these act by binding to penicillin binding proteins. The bacterial cell walls contain penicillin binding proteins, or PBPs in short, of many different types. One of the important ones is transpeptidase. Transpeptidases are important enzymes which are required for synthesis of the peptidoglycan. So these beta-lactams bind to transpeptidases, and because of which the peptidoglycan synthesis is affected and the cell will die because cell wall will not be formed. The next group are the beta-lactam combinations. Now we all are aware of antimicrobial resistance, and one of the important categories of antimicrobial resistance in bacteria are those bacteria which make beta-lactamases. Now beta-lactamase is an enzyme which breaks the beta-lactam ring of penicillins, that is of the beta-lactam antibiotics. So obviously the beta-lactam antibiotics would not be effective against beta-lactamase producers. So we can use combinations of two antibiotics which work in two different ways. So in beta-lactam combinations, we have a beta-lactam antibiotic which is having the actual activity that is going to bind to the transpeptidase. So those are ampicillins and amoxicillins, for example, and that is combined with another beta-lactam antibiotic which does not have actual activity, but what it is doing, it binds to the beta-lactamases. So these antibiotics like sulbactam and clavulanate, they bind to the beta-lactamases, and thereby it prevents the beta-lactamase from breaking the beta-lactam ring. So these combinations of ampicillin with sulbactam or amoxicillin with clavulanate are very often used for treating infections. So they are effective against beta-lactamase producers also. The third class are glycopeptides. Under glycopeptides, the very well-used antibiotic is vancomycin. We all know about how SORS is becoming resistant to many antibiotics, and vancomycin is often used for treatment of these kind of resistant SORS infections. Now vancomycin binds to precursors of the cell wall. So during cell wall synthesis, precursors are made inside the cell and then they are transported outside the membrane and assembled over there. So vancomycin prevents the binds to these precursors and prevents cell wall synthesis. Now disadvantage of vancomycin is that vancomycin is a very large molecule, and because of which it cannot penetrate into the outer membrane of gram-negative cells. Gram-negative cells, in addition to peptidoglycan, they contain an outer membrane, and vancomycin is not able to go through the outer membrane. So obviously it is not effective against gram-negative bacteria. It can work only on gram-positive bacteria, example Staphylococcus aureus. Coming to the next group, that is antibiotics or antibacterials which damage cell membrane or prevent cell membrane formation. So they damage the bacterial membrane, because of which the membrane will develop pores and all the essential cellular contents will come out, and obviously the bacterium will die. Under this you have examples like lipopeptides, polymyxins and polypeptides. So lipopeptide, the very common example is daptomycin. Daptomycin binds and disrupts the cell membrane, but only of gram-positive bacteria. So cell membrane is an important structure for the bacterium, we know that, and if the cell membrane is damaged, if it develops pores, then obviously the cell is going to die. The second group of cell membrane inhibitors are polymyxins. Under this, examples are polymyxin B and colistin. They also disrupt the cell membrane, and because of which there is leakage of important macromolecules in the cell and ions which will leak out of the cell, and the cell will die. But these polymyxins are more effective against gram-negative and not much against gram-positive bacteria. The only disadvantage of polymyxins is that they can be toxic to humans also. Now, we humans also contain a cell membrane which is similar to the membrane of bacteria. So, polymyxins can be toxic to humans because the human cell membrane may also be damaged. So, they are usually used as a last resort. Then we come to antibacterials which affect the protein synthesis. That is, they affect this translation step. They may affect the ribosome, either 30S or 50S ribosome, and they prevent translation from happening. So they disrupt protein synthesis. Proteins are very important for the cell, and if the protein synthesis is affected, obviously the cell is not going to survive. So under this we have examples like tetracyclines, aminoglycosides, very popular common example is streptomycin, the macrolides, and lincosamines, etc. So under aminoglycosides, we have examples like gentamicin, streptomycin, kanamycin, and amikacin. These are very commonly used antibiotics. How do they act? Now we know that the ribosome machinery consists of two parts of the ribosome, that is the 50S ribosome and the 30S ribosome. So these antibiotics bind to the smaller ribosomal unit, that is the 30S ribosomes. And since they bind to the 30S ribosome, they interrupt the reading of the mRNA. In translation, we know that the mRNA is read by the ribosomes. Reading means the mRNA information is converted into protein. So they interrupt this mRNA reading because they prevent the formation of ribosome and mRNA complex. The mRNA comes to the ribosome and forms a complex as the first step. That step is disrupted by these antibiotics. Now, since all types of cells contain ribosomes, whether it is prokaryotes or eukaryotes, gram-negative, gram-positive, so that's why inhibitors of protein synthesis are broad spectrum antibiotics. That is, they affect both gram-positives and gram-negatives. Now, very often, for best results, they are used along with the beta-lactam antibiotics because beta-lactams will inhibit peptidoglycan synthesis. The aminoglycosides will inhibit protein synthesis, and both together will give a better synergistic effect, a better effect or better killing of the bacteria. The next group of protein synthesis inhibitors are the tetracyclines. They also bind to the 30S ribosomes, but they don't affect the mRNA binding. Instead, they affect the binding of tRNA to the ribosome. Now, during translation, tRNA along with the amino acid comes to the ribosome and does the protein synthesis. So, the tRNA binding is prevented by the tetracyclines. Again, they are also broad spectrum antibiotics. Third group under protein synthesis inhibitors are the MLS group. MLS stands for macrolide, lincosamides, and streptogramine. Under macrolides, we have common examples like erythromycin, azithromycin, and clarithromycin. Clindamycin is an example of a lincosamide. Now, all these antibiotics work in a common way. They bind to 50S ribosomes and not 30S ribosomes. Now, because they bind to the 50S ribosome, again they prevent the protein synthesis by preventing formation of the peptide chain, that is the amino acid linking and formation of peptides which happens. That step is prevented by these group of antibiotics, and they are more effective against gram-positive bacteria compared to gram-negative bacteria. Now, when some antibiotics are effective more against gram-negative or gram-positive or any particular type of organism, it depends on the size of that antibiotic, whether it can enter that cell and whether it can reach its target site. So, there are many factors which are going to decide whether an antibiotic is able to act against a bacteria or not. The fourth class are the chloramphenicals. Chloramphenicol also work in a similar way. They bind to the 50S ribosome and they prevent formation of the peptide chain. Next, we'll come to inhibitors of DNA and RNA synthesis. DNA synthesis, in other words, is replication. So all these antibiotics, the fluoroquinolones, many antibiotics come under this category. They prevent the formation of DNA. That is, DNA is formed during replication and during repair. So that important step of the cell cycle is prevented or disrupted by these class of antibiotics, by this group of antibiotics. The second group of antibiotics affect RNA synthesis. RNA synthesis, in other words, is transcription, which is also a very important part of the cell, a part of central dogma of the cell, one of the steps. And RNA synthesis is prevented, therefore the cell is not going to survive. So under inhibitors of DNA synthesis, we have the quinolones or the fluoroquinolones. Under this, the examples are nalidixic acid, ciprofloxacin and ofloxacin. Now, how do they act? Now, these antibiotics, the fluoroquinolones, they bind to the DNA gyrase. Now, DNA gyrase is a type of topoisomerase 2, which is very important for coiling or supercoiling of the DNA. Now, when DNA replication is happening, ahead of the replication chain, supercoils are formed, and DNA gyrase helps to relax those supercoils. So, if DNA gyrase is not functioning, supercoils will build up, and therefore replication will halt and the cell will die. So, these antibiotics act on DNA gyrase during replication. Metronidazole. Metronidazole actually damages or breaks the DNA strands. Now, interesting thing about this antibiotic is that it is activated only when it gets reduced. And how does it get reduced? It undergoes reduction under conditions of low redox potential. It means that it gets activated in an anaerobic bacteria. So it is mostly effective against anaerobic bacteria and under those also against gram-negative anaerobic bacteria. Why? Because it is activated under reducing conditions. The third group are the rifampicins. Now, rifampicin inhibits RNA synthesis because it prevents the action of DNA-dependent RNA polymerase. Basically, it is preventing transcription. So, it prevents synthesis of all kinds of RNA, including the primer. You know that primers are required for DNA replication. So, it prevents the action or functioning of the RNA polymerases. But again, with rifampicin, it is not effective against gram-negative bacteria because it's a big molecule. It cannot penetrate or pass through the outer membrane of gram-negatives. So it is effective only against the gram-positive bacteria. Finally, we come to antibiotics which affect other pathways. So we are going to take example of one pathway, that is the folic acid synthesis pathway, which is an important growth factor required for the growth and multiplication of the bacterial cell. So in this we have three classes. First are the sulfonamides. Now sulfonamides disrupt the folic acid pathway by binding to an important enzyme called as dihydropteroate synthase. Now because of this inhibition of this enzyme, folic acid is not formed and therefore the bacteria will die. This is again effective against most bacteria because bacteria, that is its broad spectrum. However, it is not effective against a very important pathogen, that is Pseudomonas aeruginosa. Trimethoprim also acts on the same pathway. However, it binds to another enzyme, that is dihydrofolate reductase. So both these sulfonamides and trimethoprims act on the same pathway, that is folic acid synthesis, but both of them bind to two different enzymes. So both these antibiotics, sulfonamide and trimethoprim, are always used in combination so to get the best action against bacteria that they are trying to treat. So both these antibiotics work in synergism with each other. So they are affecting the same pathway by inhibiting two different enzymes. So they are always used in combination. And finally we come to nitrofurantoin. Now the exact mode of action of nitrofurantoin is not known, but it can target many sites like protein synthesis, enzyme synthesis, and also it may damage DNA. So it has multiple targets within the cell. However, this is used only for treatment of urinary tract infections, so not commonly used for any other types of infections. So we have seen that how there are so many multiple types of antibacterial agents which can act on different important processes in the cell, different important structures within the cell, and thereby they can give the necessary antibacterial action. Thank you.